Good afternoon, everyone. I'm Jane McIntosh. I'm the Chief Philanthropy and External Affairs Officer with the Alzheimer's Drug Discovery Foundation. And today I'm joined by our co-founder and Chief Science Officer, Dr. Howard Fillit. And we're incredibly excited to be with you all today to talk a little bit about highlights from 2024 and also to look forward to what's to come in 2025. For those of you who are new to the Alzheimer's Drug Discovery Foundation, a couple of things to note. We were co-founded by, excuse me for 1 second. I'm just going to pause and let our communications team know that we are able to hear them and ask that they mute. Thank you. So the ADDF was founded by Leonard and Ronald Lauder, along with Dr. Fillit, in honor of Estee Lauder, who had Alzheimer's disease, and Howard was actually her physician. And I bring this up because, of course, our origin story is important, but also because we were started 27 years ago, and a lot has happened in more recent years with Alzheimer's research. So Howard, just thinking about where we are now, we have new disease-modifying drugs. We've got some new tests available on proven steps to prevention. Can you talk a little bit about, did you ever think this moment would come, and what has been the ADDF's role in advancing the science?
Well, I think we played a critical role. Our three pillars were biomarkers, or diagnostic tests, but primarily to develop the biomarkers so that we could do better clinical trials. And I think that's really what proved to result in the first disease-modifying drug getting what was called at the time, a couple of years ago, the accelerated approval, which was based on the brain scan that we helped to develop starting about 25 years ago. So we played a critical role in the first brain scan, the amyloid brain scans that were used not only for diagnosis in the clinic, but also to recruit patients into clinical trials who actually had Alzheimer's disease. The other critical role we played was going back more than 15 years ago, we started investing in blood tests. Yeah. And so 2024 has been called kind of the year of the blood tests because we have several blood tests that are coming to market in various stages. Fujiray Bio is actually going to the FDA for the first approval of a blood test. So a company called C2N that we invested in about 15 years ago and continue to invest in has a blood test that will be widely available and is considered the most accurate test. And another company called Alpath, which developed a monoclonal antibody that is used in diagnostic tests by several organizations. And those tests are going to replace, we think, the brain scan. So that'll be another major breakthrough.
Can you talk a little bit more specifically to the science of each one of these tests, beginning with C2N? How does it work? What are they measuring?
So C2N is based on a technology called mass spectrometry, which is really a way of measuring individual molecules in a sample using various sophisticated physics and chemistry and so on. And so what C2N has been able to do is use mass spectrometry to identify beta amyloid in samples, even small, tiny changes in beta amyloid, to detect various forms of beta amyloid that are specific to Alzheimer's disease, and also tau tangle molecules, tau being one of the major components of the tangles that we see, which are representative of dying and degenerating neurons. And so we can actually pick up very specific chemical forms of tau, and they represent different, they have different characteristics. So there's something called p-tau 181, p-tau 217, and so on. And so in many ways, using the mass spectrometry technology, C2N has developed the most sensitive and accurate test. The problem with it has been the wide availability of these instruments. And so what we're currently doing is we've invested in C2N to help them make an instrument that will be widely available and be able to compete in the marketplace with the best test out there.
And so what would be the difference then between, say, a test by C2N versus Alpath or Fujirebio's Bio's test?
At some levels, they're similar. Fujirebio's Bio's is really based on an immunological assay.
Mm-hmm.
And so the sensitivity of it is not as great as a mass spec test. And the Alpath, that company that we invested in, makes the reagents, the monoclonal antibodies that bind to a specific type of beta amyloid or tau. And because it's the best monoclonal for the immunological assays, it turns out to be a very valuable asset that's being used by companies like Lilly and others to develop their own assays and put those on the marketplace.
And Alpath, talk about that test.
Well, that was the test I just actually.
Oh, excuse me. I'm sorry. Fujirebio Bio.
Fujirebio. So they have a technology called Lum Pulse,
okay,
which is an immunological assay, but it's a very sophisticated, sensitive assay. But they still need the reagent like Alpath has to be able to run the assay, basically. So it's immunologic assays are just not, at least at this point, not as sensitive as mass spectrometry and other technologies that are coming closer to marketplace.
So would you envision then, say, 10, 15 years from now, someone like me, I'm in my 50s, that this would just be part of my, annual wellness visit with my doctor? Would this be something that's available for the masses, that just be part of your standard annual physical?
I think there'll be several uses for this. The main one I can see right now is happening now.
Yeah.
Which is if somebody comes into the office, let's say a primary care doctor, to see their primary care for a visit for hypertension or whatever, an annual wellness visit, and they have memory complaints. Mm-hmm. So one thing the doctor can do now in 49 states, except for New York, which we hope will come in the coming weeks, actually, an approval, that the doctor will be able to do one of these tests, the blood tests that we just talked about, and confirm whether or not the patient has Alzheimer's disease. The thing about it is that we know now, and from data in the last couple of years, that the blood test correlates really well with the brain scan. Mm-hmm. So the brain scan is expensive, it's a little bit invasive, it's kind of time consuming. The blood test is just part of routine care. So the world is changing rapidly, and I think in 2025, we're going to see a lot of uptake and practice with these blood tests in clinical care. And I think the primary purpose will be to detect the presence of Alzheimer's disease in people with memory complaints who come to the primary care doctor or to a neurologist that they've been referred to. And I think that'll be the main use. It's also, as you mentioned, Jane, used in screening. Mm-hmm. So people who are, since we know the disease starts like 20 years before there are symptoms, and people who want to know what their risk is, they'll be able to go to the doctor and get these blood tests and see if they actually have preclinical Alzheimer's disease. And where that's really going to be important going forward is in prevention, because we're going to be able to detect people before they have symptoms and put them into prevention programs that, based on studies by doctors like Dr. Kivipelto out of Scandinavia, which her study being replicated now in over 60 tests around the world, we know that prevention works. And so if we can pick up people early, even before they have symptoms, let's say they have a strong family history, mother had the disease and grandmother had the disease, and they want to know if they have that risk, one of the things they'll be able to do is get a blood test.
Can you talk a bit more about Dr. Kivipelto's work and prevention generally? Her finger study and the lifestyle modifications that we now know are critically important for people and their brain health.
Yeah, it turns out that preventing brain disease, Alzheimer's, neurodegenerative disease, just like preventing heart disease, let's say, is very similar. So you have there are 12 major factors, but there are sort of 5 of the big ones. Mm-hmm. One is if you have diabetes or hypertension, you really want to control those because those are risk factors for cognitive decline and also for dementia, including Alzheimer's disease. You want to stay socially and occupationally engaged. You want to exercise. Now, that doesn't mean you have to be a star. Like you can exercise for 20, 30 minutes 4 or 5 times a week. The general recommendation is to try to get a moderate amount of exercise, and it could be strength training, it can be aerobic training, but about 150 minutes a week, which would be about 30 minutes, 5 days a week, or something like that. Walking is fine, brisk walking is better, but exercise turns out to be really interesting, not just because it works, but also because of the biology of how exercise actually does work to prevent cognitive decline. And the other thing is remaining socially and occupationally engaged, because people that become isolated socially, actually, it's kind of a use it or lose it situation. What Dr. Kiva Pelto did was she actually did a randomized trial, which is the highest order of evidence in science. And she put one group into sort of a placebo group where they got the information about these 5 major risk factors, but they weren't in any sort of compliance program to make sure that they did it. And that was kind of your placebo group. And then she put the other in an experimental group where they actually came to the clinic on a frequent basis to make sure they were being compliant with these recommendations, that they were exercising. She gave them diets, food, to make sure, as Dr. Ornish did out in California to replicate this, so that to make sure that they actually complied with the diet, they got the right diet. So the experimental group actually got those 5 interventions, and there was a program to make sure they were compliant with it. And the people that just were in the control group just got the recommendations and were left to their own. And it turned out the people who actually were compliant had a slowing of their cognitive decline, even if they had mild cognitive impairment. And what we're doing now is sort of finger study 2.0, where we're adding metformin, which is the leading anti-aging drug, and in some studies has been shown to be beneficial in people with mild cognitive impairment, studies that we funded that are now, the principal investigators just gotten about a $30 million grant to do a large trial of metformin in people with very early Alzheimer's disease. So we're combining, just like in heart disease, we do those lifestyle interventions, and we add a statin in brain degeneration with aging, brain aging, and Alzheimer's, early Alzheimer's. We're going to be doing the lifestyle interventions, management of vascular risk factors like hypertension and diabetes, and adding metformin.
So I want to pivot slightly and go back to the newer drugs that are now available. Eli Lilly's drug Kisunla got FDA approval last year. That's a major watershed moment in Alzheimer's research. Can you talk a little bit about this drug, how it works?
Sure. So, of course, people with Alzheimer's are defined by having plaques and tangles in their brain. And we know that these beta amyloid molecules that are predominantly the major molecule in these plaques, that they're neurotoxic in all its various forms, the beta amyloid. So what we want to do, there's been various attempts to remove or prevent the deposition of beta amyloid in the brain, kind of inhibiting enzymes that lead to the formation of beta amyloid from a larger molecule. Those worked in some degree, but they were very toxic. These were called beta secretase inhibitors, going back maybe 20, 25 years. So the other thing is, well, once the plaques are formed, they're toxic. What about trying to remove the plaques from the brain? And there's been a lot of attempts at trying to do that. One thing that happens is the immune system is critical for removing the garbage that is the result of metabolism in all cells. And we think that the plaques are kind of these little pockets of molecular garbage that deposit in the brain and cause neurotoxicity. So the way drugs like the monoclonal antibodies, like Kisunla and Leqembi, is that they are immune molecules, they're antibodies that bind to the plaques and say to the cells that remove plaques, that are immune cells that remove the garbage, we're here, there's garbage here, you have to remove it, and specifically beta amyloid. So these are monoclonal antibodies. It means that they're immune antibodies that are very, very specific to beta amyloid, and they see this form of beta amyloid in the plaques. They bind to it, and a piece of the part of the antibody that sticks out says to these immune cells, come over here, there's stuff here you got to get rid of. Right. And that's how they work, basically. And they work quite well. And we know they work well because of the PET scan that we helped to invent. Because in order for people to get into the trial, they have to have a positive amyloid PET scan. And then that PET scan is used to show that the antibodies work, because we wait, especially with the Cuna, which is the Lilly antibody, that people come off the drug when the PET scan turns negative. And so it's proven through the PET scan in clinical trials that the drug actually removes all the beta amyloid, as far as we know, that we can see with the PET scan from the brain, which is really quite an amazing advance. And the last part of the piece is that there's been this amyloid theory for many years, that the plaques and the beta amyloid play a role in neurodegeneration and progression of the disease. And so the correlation with the negative PET scans, the PET scans turning negative, indicating that the antibody, the drug, has gotten rid of the PET scans, is that people decline at a slower rate. So there's a clinical benefit. And so that kind of fills all the requirements for a drug to be deemed as effective.
Okay, I'm going to pause for just 1 second, and I want to just encourage those who have questions to please feel free to submit. We have a moderator on the other side who's going to be texting them to me so I can ask them for the next 20 minutes or so. I have a follow-on question. So we here at the ADDF have long pursued an approach to our investing that really looks at the biology of aging, how a person ages. And so therefore, this one drug or this new suite of drugs, it would seem it's not the full picture in terms of what a patient with Alzheimer's will ultimately pursue in the clinic. So can you talk about where this is all going and what you see the future for treatments?
Right. So for one point, we know that removing the plaques slows the disease down by 30, 35%. It doesn't encompass 100% to slow it down. So there must be other factors. Second thing is aging is by far the leading risk factor for Alzheimer's disease. So what we haven't done in the field very well is translate almost 100 years of research on aging and understanding 8 or 9 pathways of aging that are the predominant, what we call the hallmarks of the biology of aging, and translate that knowledge into new drugs for Alzheimer's disease. The same way that we've been able to recognize that aging plays an important role in cancer, right, and using that knowledge to develop various cancer agents, some of which we've actually repurposed for Alzheimer's disease. A good example of aging-related biology and Alzheimer's is inflammation. Mm-hmm. And we know that one of the really major hallmarks of aging is inflammation. We've known that inflammation plays an important role in Alzheimer's disease for decades, but what we haven't been able to do is translate that into effective therapeutics to actually halt the brain inflammation that we think not only contributes to neurodegeneration, but probably increases the amount of amyloid and plaque formation, and ultimately toxicity indicated by the tangle formation, which is a sign of the tangles that are leading to neurodegeneration and actually death of the neurons. So, non-steroidals like Motrin have been tried for Alzheimer's. They didn't work. Mm-hmm. Other anti-inflammatories have been tried. But now we're in a new age of immunology as well. And so using some of the most modern immunologic agents, companies like Coya Therapeutics, mm-hmm. Which is a company we've invested in, is attempting to approach the immune dysregulation, which is occurring, and the immune component, the inflammatory component of Alzheimer's. And I think there's some expectation and possibly even confidence that by re-regulating and normalizing the immune system and inflammation in Alzheimer's, that we'll be able to capture, along with removing the plaques and maybe the tangles, a better way of slowing the disease down and maybe even preventing it.
That's incredible.
Yeah, it really is, because I think, I have to admit that when I started researching 45 years ago,
yeah.
I really actually was very interested even back then and published back then because I had trained as an immunologist. I came to it with that bias or that perspective and did publish like 40 years ago and 30 years ago some research. And it was just an example of an idea that was before its time. It really wasn't well accepted. But now things have come around again. And I think we're seeing a lot of interest in inflammation. Of the non-amyloid, non-tau approaches, inflammation is probably the largest bucket of new drugs in development.
That's pretty extraordinary. I mean, just to see how the field has shifted in this period of time. I know you've talked about how, Alzheimer's research is kind of where cancer research was about 30 years ago, and that tends to be about the horizon for this kind of a work. A couple of weeks ago, we were in Palm Beach doing a program with another expert, Dr. Nir Barzilai out of Einstein. And he's a longevity specialist, and we had a very spirited discussion with him. Can you talk a little bit about just this field of longevity science and how it relates to Alzheimer's disease?
Well, for one thing, I'll give an example. There are cells in every organ in our body that are called senescent cells.
Right.
And they're kind of, in a way, we don't completely understand it, but they're kind of, in a way, cancer cells because they don't die. So cells have a way of dying because when they get sick, they're kind of getting in the way of organ function. And I'm kind of doing a simplistic point of view about this. But these senescent cells that don't die for reasons we don't completely understand are very inflammatory. So in the brain, we see senescent cells that don't die. So one thing we want to do is, we don't think we can re-regulate them, but we do think that we can help them to get out of the way and die. And so one of the things that we're funding right now is a clinical trial at Washington University in St. Louis with Dr. Miranda Orr to fund the use of a cancer drug that helps cells to, like senescent cells, to die, along with a flavonoid, which is a combination therapy. So it's a drug called dosinetab with quercetin. This combination has been shown to help senescent cells to die and reduce inflammation. And so we're adopting that kind of cancer approach to the treatment of Alzheimer's disease in a clinical trial. And it's one of the first clinical trials of this approach, which is really a new class of drugs called senolytics. And these are the first kind of senolytics to get into clinical phase testing. And it's an example of really a very specific aspect of the biology of aging being applied to Alzheimer's therapy.
Gotcha. Can you talk a little bit more about combination therapy? And do you see it as sort of one monoclonal antibody or multiple, or how do you think this is going to play out?
Well, I want to say, I think combination therapies with monoclonals will be hard to do. They require the infusions. They're a little bit expensive, but not extraordinary at all for drugs of its class. I don't want to make it seem like they're overly expensive. They're not. They're within the range of these kind of biologics. But what we expect to happen in the next year is that there'll be new forms of administering the monoclonal antibodies, particularly subcutaneous administration is in development, and I think will be applied for in the very near future, if it hasn't already been in the last few weeks, that the FDA will approve an injectable. So patients will be able to take these drugs at home. And if that happens, and I think it will, then we'll be able to much more easily and efficiently use the monoclonals in combination with other drugs. So imagine using a monoclonal antibody in a subcutaneous injectable, maybe once a week, alongside a drug like a GLP-1 agonist like semaglutide, which has not only anti-obesity properties, but has been shown to be valuable for chronic renal disease and heart disease. And now we're seeing evidence that we supported several years ago of this class of drugs called GLP-1 receptor agonists as beneficial in people with Alzheimer's disease. We supported a precursor drug to semaglutide, which is Ozempic in the United States, a drug called liraglutide at the Imperial College of London, which incentivized, we think, Novo Nordisk to do an ongoing clinical trial of Ozempic or semaglutide for people with Alzheimer's. And we expect the results of that trial in the coming months. If it works, then that would be a natural drug for a combination therapy of a GLP-1 receptor agonist along with a monoclonal antibody. But there are many iterations of how these combination therapies can be designed and developed, and which combinations and so on. I think it's going to be many years of doing these experiments to find the best combination and find the best protocols and clinical trial designs for doing combination trials. But let's not forget, in cancer, combination studies and combination drug administrations are really the state of the art, because it's kind of a little bit like whack-a-mole. You hit one target, but the other one comes up and the cancer survives. You hit 2, 3, maybe 4 targets, and you can able to treat the cancer. And cancer is a disease of aging primarily, and there's a lot of similarities. So we do think that precision medicine, getting the biomarkers, characterizing a patient's brain biomarkers, and then tailoring a combination therapy for that person is the future of treatment for Alzheimer's and related dementias, for that matter.
All in our wheelhouse, Howard. That's a good thing. This has been a delightful conversation. I'm going to wrap this up, but I want to bring up a couple things to our audience here. Howard is going to be participating in a special webinar on January the 29th. He will be joined by Eli Lilly's Laura Steele, the Dementia Discovery Fund's Lawrence Barker, Cognition Therapeutics, Christian Howell, and Wake Forest University's Michelle Milkey in a special webinar through Endpoints. And as a follow-up to this discussion, we will send information on how to sign up for that. The discussion will be all around what's in store for 2025. I also just want to mention that we are hosting the inaugural ADDF Scientific Summit in Palm Beach on March the 5th. We have a group of 8 Goods Prize winners who are among the most innovative and accomplished scientists in the world in the Alzheimer's field, along, of course, with Howard, who is one of our co-hosts, along with Sanjay Gupta from CNN. This event will, as I said, take place on March the 5th and is open to the public, and you can find information about that on our website as well. Lastly, I'd just like to thank all of you so much for listening to us today. Apologies for our slight technical issues at the beginning, but Howard, always a pleasure to talk with you. And of course, we are so grateful to each and every one of you for your support and engagement with our work. It's really an incredible privilege and honor to be part of this organization that is advancing the field of Alzheimer's research. And we wish you all a wonderful 2025. And I'm going to make you smile. Thank you all so much.